Hello everyone. In this session, we are going to focus on one principal component of cytoskeleton, and that is microtubules. So let us look at the learning outcomes of this session. One of the principal components of the cytoskeleton, uh, which is the microtubules, it has a size of 25 nanometer and they are considered to be the largest cytoskeleton. They are rigid, hollow, cylindrical, rod-like structures. The microtubules are dynamic structures, which means we can understand it very nicely that they are assembling and disassembling, and they arise from microtubule organizing centers. So they have a nucleation center from which they are assembled. They can be stabilized by microtubule associated proteins. That means when one wants that the disassembly of the microtubule be stopped, then you have certain microtubule associated proteins that can go and bind to the microtubule and stop its disassembly. So you have means of kind of preventing further polymerization or further depolymerization that is possible. Apart from maintaining the shape of the cell, they have a major role to play in intracellular movements and cell locomotion itself. They are associated with molecular motors, which enable the microtubules to enable motility and intracellular movements. Let us look at the structure of the microtubules first. So, every microtubule is actually, as we see, a long cylindrical structure, hollow at the center. So you have this kind of circling as a helical structure. So you have every filament, protofilament that is present in it, circularizing helically to kind of form the entire hollow structure. And every microtubule present in the microtubule per se contains 13 protofilaments. So you can see the diameter of the microtubule is 25 nanometer, the inner diameter being about 14 nanometers. So you can kind of understand how the protofilaments take up about 10 nanometer of space or 10 to 11 nanometer of space, and together the outer and the inner together match. When you match it, then it is about 25 nanometers in diameter. So it is the largest cytoskeleton. But what we need to understand is that every microtubule is made up of 13 protofilaments. So you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13. So there are 13 protofilaments that make up one microtubule. That is important to note. And you can kind of, make an analogy with the fact that if you have one stick, it is easy to break. But if you have 10 sticks together, then it becomes very strong. So therefore, the microtubules also, because they have 13 protofilaments, together they make a very strong hollow cylindrical structure. Now, every protofilament is made up of what is called as a tubulin heterodimer. So there are three types of tubulins present in the cell. One is an alpha tubulin, beta tubulin, and gamma tubulin. Alpha and beta tubulin are actually the structural components of the microtubules, but gamma tubulins are not the structural components of the microtubules, but they help in the assembly of the microtubules, and we are going to look at it at a later stage. So, two monomers, that is alpha tubulin and beta tubulin, come together to form a dimer. It is called as a heterodimer because both the tubulins are structurally a little different from each other in terms of the amino acids that are forming its primary structure, and therefore as a folded structure they may be slightly different from each other. So you have one as an alpha tubulin and the other one as a beta tubulin, but together the alpha tubulin has a binding site for beta tubulin, and beta tubulin has a binding site for alpha tubulin, and that is how they interact with each other. Another important fact over here is that both alpha tubulin and beta tubulin have a domain to which you can have a nucleotide bind. So they have what is called as a nucleotide binding domain, and specifically it is GTP, that is guanosine triphosphate, that comes and binds to that nucleotide binding domain. So both alpha and beta tubulin are associated with GTP. Which brings us to another fact, and the fact is therefore that both alpha GTP and beta GTP, the alpha tubulin and the beta tubulin, have an inherent GTPase activity. So they have an enzymatic activity, that is, they not only bind to GTP but they can hydrolyze GTP to GDP. That is something that is very important. But the point to be noted is that when you have a tubulin heterodimer per se, you will not have the GTPase activity of these fully functional. They may be very, very slow and weak. Okay, but they can be accelerated. That means the GTPase activity can be accelerated when the conformation changes. So we'll look at it in a while. So when you have these heterodimers binding to each other, they form what is called as a protofilament. So this is one protofilament. Over here, you must note that you have always the heterodimer associating in this manner, that is alpha beta alpha beta alpha beta alpha beta alpha beta. You will never have alpha beta beta alpha beta. So you must understand therefore that the protofilament will always have at one end the alpha tubulin, at the other end you will have the beta tubulin. So you have what is called as a polarized structure. And also one other thing that is to be noted is that the alpha GTP tubulin end is what is called as the minus end, and the beta GTP tubulin end is what is called as the plus end. So that also is polarization. But the question that comes up over here is, why have we called this as minus n and why have we called this as plus n? We do definitely understand polarization in terms of structure. That is, at one end you will always have the alpha GTP, and at the other end you can see you have the beta GTP ended. So if you have more heterodimers coming in, increasing the length of the or extending the microtubule, you will always have the alpha present here and the beta present here. So you will always have at this end the beta tubulin, and at this end you will always have the alpha tubulin. So that makes it structurally polarized. But you also have what is called as minus and the plus end. So what does that mean basically? It means that the plus end is the fast growing end of the microtubule. So if the microtubule has to grow in length, then the polymerization is more at the plus end. So you will have more and more heterodimers coming and binding at the plus end and extending the microtubule. In comparison, at the minus end, it will be much lesser. That means you may have the heterodimers coming and binding over here, but the rate at which it binds at the minus end is much, much less. Okay, and so therefore this has been termed as the minus end because the growth is slow at this end, and this end is called as a plus end because the growth is more at this end. Here you need to keep or here you need to take into account one more thing, and that is when you have the when you have the alpha beta alpha beta alpha beta kind of growing on each other, okay, and extending the microtubule. One thing that has been observed is that when the next heterodimer comes and binds, so you have this heterodimer, and when you have this heterodimer coming and binding, the binding of alpha GTP to the beta GTP is going to bring in a conformational change in the beta tubulin. And this conformational change activates its GTPase activity, and therefore the GTP that is bound to the beta GTP is going to hydrolyze to form beta GDP. So therefore you can see when you have the protofilament, the growing end will always be beta GTP because you don't have an alpha tubulin yet coming and binding. But here, because the alpha tubulin has bound to the beta tubulin, the beta tubulin's GTP has got hydrolyzed to GDP because the alpha tubulin has brought in a conformational change in the beta tubulin to activate its GTPase, and that GTPase is able to convert the GTP into GDP. What is the outcome of that? The outcome of that is that this beta GDP would have a less affinity to bind to the next heterodimer. So its affinity to bind to the heterodimer is decreasing. But then the point is that the energy that is released is able to kind of hold on to these heterodimers because the next one will come in immediately. So you would have polymerization happening as the alpha beta dimers are going to come and bind to this end. but every beta tubulin would have got converted to its GDP form. So you will have alpha GTP, but the beta will be GDP. You have alpha GTP, but the beta would be GDP. So that is how it is growing. Only at this end, that is the plus end, the dimers that have come in, those dimers, the GTP is still intact. Question: for how long? Because the beta tubulin has a weak GTPase activity. Even if you do not have any more heterodimers coming and binding, after a point you will find that this beta GTP is getting converted to beta GDP, and essentially you have energy being released, and that energy being released can lead to these heterodimers being released. We will just see in a minute. So let us understand what we have tried to kind of look at in terms of the structure. When you look at longitudinally, when you look at cross section, you will find that every microtubule is made up of 13 protofilaments, and every protofilament is made up of a polymer of alpha beta heterodimers, and it is aligned in such a way that you will always have the alpha end at one side, and you will have the beta at the other end. So you have a polarized structure, plus you have a minus end wherein the growth is much smaller or much slower, while you have the plus end at which you will see that there is a lot of tubulin dimers coming and associating. And so at this end you will have what is called as a GTP cap. Why? Because only when you have the next dimer coming and binding, this GTP associated with beta tubulin is going to get hydrolyzed fast. But if you don't have beta tubulin, if you don't have the next dimer coming and binding, after a point of time, this GTP will hydrolyze on its own, because the beta tubulin has its GTP as active but very, very weak. So when, I'm repeating again, when you have alpha bound to the beta tubulin GTPase activity is very enhanced. But when you don't have the alpha coming and binding to the beta tubulin, its GTPase activity is there, but it is very weak. So it will not immediately get hydrolyzed to GDP. It will get hydrolyzed after a certain duration, because the activity is very weak. So it will hydrolyze, but it will hydrolyze after a certain time. So that is something that one has to keep in mind. Now, therefore, since we looked at how the microtubules are being formed, you can see how it is getting polymerized, but let us also look at the fact that it can get depolymerized. So assembly and disassembly of the microtubules is what we understand by dynamic instability. So let us say, for example, in the cell, there is sufficient amount of tubulin dimers. So you have sufficient amount of alpha beta tubulin dimers, and interesting point that you must keep in mind over here is generally within a cell, the concentration of the dimers, okay, is almost in equilibrium to the dimers that are present in a filament. So understand, the dimers that are present in the filament is in equilibrium with the dimers that are not yet part of a filament. Okay. So here let us take into consideration that there is in the cell sufficient amount of the dimer present, that is the heterodimer present. And so what is going to happen is at the plus end, you would have these heterodimers coming and binding to the alpha tubulin. So the the alpha tubulin of the incoming heterodimer is going to bind to the beta tubulin of the existing layer. Okay. So on binding, you can see how immediately you will have GTP getting converted to GDP and the PI being released. So this is associated. Then you have the next set coming, and that is how you would have the polymerization happening, extending the length of the microtubule. So what is this? This is basically polymerizing microtubule. That is, the microtubule is growing. Okay, now you must understand that it can grow at this end also, but here what we are generally showing is that it is growing much faster at the plus end. So this is what we are kind of understanding when we say it is plus end. So you may have the dimers also come and bind over here, but here the rate is much smaller or much lesser. I'm sorry for using the word smaller, much slower compared to the the assembly at the plus end. This is what is shown. Now let's take into consideration that as polymerization continues to happen, at a point of time, the amount of heterodimers decreases in the cytoplasm. So if the amount of heterodimers are decreasing in the cytoplasm, once you have the alpha beta tubulin coming and binding, and you have the hydrolyzing of the beta tubulin of this layer, okay, but this over here is 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 having what is called as a GTP cap, because the beta tubulin over here has GTP still bound to it. Why? Because you do not have any further heterodimers coming and binding. But like mentioned earlier, this beta GTP will not remain beta GTP for long, because it has a weak GTPase activity. It is going to convert its GTP into GDP, and that conversion of GTP into GDP is going to release energy. And that energy that has been released will be sufficient enough to destabilize the interaction that is there between this existing heterodimers, you see. So what is going to happen, therefore? You will have these heterodimers being released. So effectively what we understand is that it is at the plus end that the depolymerizing is also happening. And so understanding is that since assembly at the plus end is fast, so is the depolymerizing. So disassembly will also be faster at the plus end, and compared to that at the minus end, the disassembly is also going to be slower. But you can understand how, because of a lesser amount of heterodimers being present, you don't have more heterodimers coming and binding over here. So what is happening in the cell is there is now no equilibrium between the heterodimer concentration and the heterodimers that are present in the filament. And so to bring it back to equilibrium, what is going to happen? The heterodimers in the protofilaments are going to be releasing themselves. So disassembly will happen, and slowly you will again have equilibrium between the concentration of the heterodimers in the cytoplasm with respect to the concentration of heterodimers in the filament or in the microtubule. So therefore, this is what we understand by dynamic instability. That is, to maintain an equilibrium between the concentration of the heterodimers and the dimers that are present in the microtubule, you will have constant assembly and disassembly. So when you have large numbers of the heterodimer present, you will have polymerization happening. And when you have low levels or hardly or negligible amounts of the heterodimer present, then you will have the microtubules disassembly assembling to ensure that the number of heterodimers in the cytoplasm again increases, so that there is now equilibrium between the microtubule formed, assembled microtubules, and the disassembled microtubules. So this is what we understand by dynamic instability. You must understand that the GTP hydrolysis weakens the binding affinity of tubulin for the adjacent molecules, and therefore it favors depolymerization. So when there is no longer any heterodimers coming in, then you would have disassembly happening, because the heat generated due to GTP getting hydrolyzed to GDP is sufficient to weaken the bonds between the existing polymerized microtubule. So it becomes, or so it begins the depolymerization. So you can see how the GTP cap is a point of regulation for assembly and disassembly. That is something that is important to keep in mind. Now, in terms of dynamic instability, therefore, one sees what we understood by dynamic instability. So for many microtubules, what has been observed is that the minus end, you do not have really assembly and disassembly happening, because it may be bound to something. So at the minus end, there is a pause. And of course, what you observe is, because of low levels of alpha beta tubulin, you may have the plus end at the pause position, but because but when you have enough number of alpha beta tubulins present as a heterodimer, you will see that at the plus end there is growth. So you have the microtubule growing at the plus end. When the levels of microtubules decrease, when the levels of the heterodimers are decreasing, you would have polymerization happening at the plus end itself, and this depolymerization is called as catastrophe. Okay, where you have assembly, from that end you will have disassembly also. And with the disassembly, what is going to happen? The microtubule is going to shorten. So with polymerization, microtubule is going to become longer and longer, and with depolymerization, the microtubule is becoming shorter and shorter. So therefore, based on requirement, you will have a certain microtubule growing in length, but you will have another microtubule probably becoming shorter. And so you can understand that the shorter microtubule is providing the heterodimers for the growth of another microtubule. So you can see how that equilibrium is helping certain microtubules to grow and certain microtubules to get shortened. And that is important in terms of functioning of the cell. Well, you also have something that is called as a treadmilling effect. So in treadmilling effect, what you see is that if you have addition of heterodimers at the plus end, then from the minus end, you will have the disassembly happening. So you have assembly here, disassembly here, assembly here, disassembly here. So what you see over here is that you have the microtubules extending from one end, and from the other end it is becoming shorter and shorter. So this is what is called as a treadmilling effect. In many cases what has been observed is that you have assembly, you have addition, and you have also removal. Addition, removal, addition, removal, addition, removal. So you have a constant turnover happening with respect to the microtubules per se. So that is what is called as a treadmilling effect, and it has been observed with respect to microtubules. Now, interesting and very important and pertinent is the fact that one can inhibit the polymerization of microtubules and one can inhibit the depolymerization of microtubules. So say for example, you have colchicine, which is a secondary metabolite obtained from plants. So it is a phytochemical that is present in plants, and this phytochemical, when you add this to a dividing cell, you will find that the colchicine is able to stop the polymerization of microtubules. So you will not have the microtubules being formed, but it is not stopping the depolymerization. So the microtubules will continue to shorten, and after a point of time, what you will find is there will be hardly any microtubules remaining in the cell, because depolymerization is happening but polymerization is not happening. So the equilibrium that is there between polymerizing microtubules and depolymerizing microtubules is lost because of the presence of colchicine. So colchicine in a way therefore can arrest the cell division. Because see, point to be noted over here is that during cell division, the spindle fibers have to be formed. So polymerization of the spindle fibers have to happen. Suppose you add colchicine and the spindle fibers are not formed, will you have the cell division progress? No, because the spindle fibers are not binding to the chromosomes or aligning it so that you have a metaphase, okay, or so that you have a telophase happening. So therefore you will have the cell arresting at that point, and when you have cell arresting at that point, cell division will not go further. You have it the other way around also. Say for example, taxol. Taxol is again a phytochemical that is extracted from a plant which is called as Taxus brevis or Taxus species of plants. They are trees, okay, and the taxol that is derived from these trees, they are able to inhibit depolymerization. So here what is going to happen is that the microtubules will polymerize and form the long extended microtubules, but it is not going to depolymerize. So you will not have shortening of microtubules happening, you will only have lengthening of microtubules happening and they remaining stable as a microtubule. So in such a scenario also, it is going to affect cell division. How? Because once you have the spindle fibers formed and they are going to bind to the chromosomes, for the chromosomes to be separated and brought to the poles, the microtubules have to shorten. And if the microtubules are not shortening, you will immediately have the cell division getting arrested. So colchicine and Taxol are used as chemotherapeutic drugs in cancer treatment. Because what is cancer? Cancer is nothing but unregulated division of cells. So you want to stop the division of cells, then you can add Taxol. The moment you add Taxol, it means that the dividing cells will get arrested and you will not have multiplication of the cells happening. So you are kind of preventing the cancer from growing by adding the Taxol. So that is therefore something that is important to note, and this therefore understand that we, having understood that polymerization and depolymerization of microtubules happening, you know when to add a inhibitor for depolymerization, and you know when to add a molecule for inhibiting polymerization, and thereby you can use it to you can use the microtubule as a therapeutic target for treatment of certain diseases. So that is extremely important to note. Now, in certain cases, once you have the polymerized microtubules, you would probably want them to remain polymerized for particular functionalities. So, to stabilize microtubules, there are certain proteins that are called as microtubule associated proteins. So when microtubule associated proteins bind to the microtubules do not disassemble fast because the microtubule associated protein binds to the microtubules such that it does not allow the heterodimers to release themselves. And so this particular map, so this is an example of molecular microtubule associated protein map 2. So as you can see, it has an extended structure. This is the tail part and this is the head part. The head part binds to the microtubules, okay, while the tail part can also bind to another microtubule. So it also is able to kind of stabilize the structure such that you have microtubules associated with each other and disassembly prevented. Now, where is this useful? Say, for example, let's consider you have a neuron. And in neuron, the structure, as you can see for neuron, is very highly defined with a part of the cell forming the cell body and another part forming the axonal end or the axonal terminus with the axon extending from the cell body to the axonal end. Now, this is very important because this axonal end is required for neurotransmitters or molecules that are produced in the cell body to be transported to the axonal end and things that are being taken up by the axon to be transported to the cell body. So, effectively, the axon is basically an area of the neuron which enables transport of molecules from one end of the neuron to another end of the neuron. So you can have molecules being moved from the cell body to the axonal end, and you have molecules being moved from the axonal end to the cell body. If that is going to be a constant process, if axonal transport has to keep happening for the neuron to function properly, and why is that required? It is required because you need to transmit a signal from one neuron to another neuron. So whatever neurotransmitters are released over here, that has to bind to the next neuron, and that next neuron has to transfer it to the next neuron, and so and so forth. So axonal transport therefore is extremely important. Okay, so if you need to have axonal transport happening all the time, the microtubules within the axon are being used as molecular tracks, or what we understand as railway tracks. And on these tracks, you have certain molecules walking or moving to transport molecules from here to here or from molecules from here to here. So, if this microtubule is stable, then the transport is going to be efficient. If it is not stable, if this is assembling and disassembling, then the transport is going to be disrupted. Okay, so in order to ensure that the microtubules that have assembled in the axon remain assembled, remain polymerized, you have microtubule associating proteins coming and binding. So for example, tau is an example of a microtubule associated protein. that would go and bind to the microtubule. So it goes and binds to the heterodimers present in the microtubule and it stabilizes it. So you can have free tubulins binding and extending, but the extended portion are bound to by the tau to keep it polymerized, to keep it stable. So this is what the role of the microtubule associated proteins are. So let's say, for example, you have a healthy neuron. So in a healthy neuron, the tau binds to the microtubule and keeps it stable. Okay, so when it keeps it stable, you can have this being used as a railway track properly, and molecules walking on this microtubule to transfer components from one part of the cell to the other part of the cell. But say, for example, the tau that is formed is misfolded, or it is not properly, or it is not enough conformation that makes it biologically active. Then what is going to happen is that the tau proteins will rather bind to each other to form a tangled mass, and it will not bind to the microtubule. If it does not bind to the microtubule, you can see that the microtubules begin to depolymerize or disassemble. And so in this, therefore, the transport can happen till here, but because it is getting depolymerized, transport will not move any further. So therefore, in a diseased neuron where tau is not functional properly, is not functioning properly, you can see how you do not have the axon, you know, uh, as erect and as functional as you can see where you have disintegrating microtubules. And therefore, this is the condition that you will find in Alzheimer's, where tau, when deficient, it kind of forms tangled mass, and that can affect transmission from one neuron to the other to the other neuron being disrupted, and that leads to a lot of neurodegenerative diseases. So that is what we understand by microtubule associated proteins stabilizing microtubules. Now we go to another component associated with microtubules, and that is microtubule organizing centers. So these are kind of bodies present in the cell from which microtubules arise. Okay, so they are the nucleation centers, or they are organelles that enable polymerization or assembly or growth of the microtubules. So microtubules can emerge from mitosis. That is what the understanding is. Say, for example, those microtubules that are required for flagella and cilia, they originate from what is called as basal bodies. Those basal bodies are present just below the plasma membrane. So as the microtubules begin growing, the membrane along with the microtubules extend into forming what is called as the cilia and the flagella. Or you have microtubules that are required for forming the mitotic spindles during the cell division. So they arise from mitosis. The best example for mitosis is centrosomes. We all know that in animal cells there is what is called as centrosomes. They are organelles that are present exclusively in animal cells, and every centrosome is made up of a pair of centrioles, and that centriole pair is surrounded by what is called as pericentriolar mass. So the pericentriolar mass is a dense region, and it is dense because there are a lot of microtubule heterodimers present. There are a lot of short microtubules being formed. So it becomes a very dense region. And this that you see is a pair of centrioles, and these centrioles are always arranged perpendicular to each other. So as you can look at this electron microscope slide, you have one centriole present in this way and the other one in this direction. So this what you see is the cross section, and this what you see is the longitudinal section of one of the centrioles. So you have a pair of centrioles being present. If you see at the cross section of the centriole, you can see that it forms a pinwheel structure, and this pinwheel structure is made up of a set of uh, 9 sets of microtubules, and each set is a triplet microtubule. What do we mean by triplet microtubules? It means that one set is made up of 3 microtubules. So of the 3 microtubules, one microtubule is complete. That means it has all the 13 protofilaments, but the other two are not complete microtubules, they are incomplete microtubules. So this has 11 protofilaments, this has 11 protofilaments, but then they share two protofilaments of the previous one. So thereby this is a triplet set of microtubules, and you have 9 of them, okay, forming what is called as the pinwheel structure. So this has, and at the center you don't have any microtubule present. So therefore this arrangement is what is called as 9 plus 0 arrangement of microtubules. So this centriole becomes the base for formation of 13 protofilament microtubules. Just to understand it a little more. So for example, you have the nucleus, and just beside the nucleus you have the centrosome. You can see how from the centrosome you have microtubules growing such that it traverses the entire cell. Or let's consider a fiber. A muscle fiber is made up of several muscle cells, but these muscle cells have fused with each other to become a multinucleate structure. Again, you can see how wherever there is a nucleus close to that, you would have a centrosome which would give rise to these microtubules. And so the entire muscle fiber, you will find microtubules present. Now, uh, a lot of studies have been going on with respect to understanding what initiates the microtubule formation and what does not. So there are a lot of things, a lot of molecular mechanisms that are involved. We will not go into that depth, but what is understood is that at the surface of the centrosomes is present certain proteins which are called as attachment or adapter proteins. And to these attachment adapter proteins come and binds what is called as a uh, gamma tubulin receptor structure. So this gamma tubulin is a ring structure, and this ring itself is made up of several gamma tubulins. So they form a ring, and that ring comes and binds to the attachment or the adapter protein. So together this tubulin, gamma tubulin, so the gamma tubulin is kind of a nucleation center allowing the microtubule to be formed. So as you can see over here, the plus end keeps growing, and so the microtubule becomes longer and longer. So the plus end is the growing end. In this case, you can see there's a pause at the minus end. Why? Because it is associated with the gamma tubulins. Okay, where it free, where it not be bound to a gamma tubulin, you may have the minus end also growing, but at a much slower rate. What has been studied is that the assembly of the attachment protein and the gamma tubulin can be associated with what is called as covalent modification, and that too the phosphorylation of the respective proteins. So when it has been studied that when the attachment protein is phosphorylated, that can go and bind to the surface of the centrosome. Okay, and then when you have the gamma tubulin getting phosphorylated, then you would have the assembly or the polymerization of the microtubules beginning. So this is something that is important. So here what you see, and interestingly what has been observed is that the minus end always faces the MTOC. Minus end is not generally the on the opposite side of the MTOC. It is always plus end which is at the opposite side of the MTOC per se. The gamma tubulins, as mentioned, form rings that become nucleation centers for microtubule growth, and it is the gamma tubulins that preferentially bind to the alpha tubulin, and since they preferentially bind to the alpha tubulin, the negative or the minus end is always found towards the MTOC, because the gamma tubulin preferentially binds to the minus end. So therefore, you can see that it is the gamma tubulins that is establishing the polarity of the growing microtubule, minus being towards the MTOC and the plus being away from the MTOC per se. Now we come to another set of molecules that is associated with microtubules, and that is what is called as molecular motors. The reason why they are called as molecular motors, we all know what a motor is. Motor is something that is able to generate energy, and the chemical energy can be converted to mechanical energy. So there is kinetics involved with these molecules. So the molecules can move because it is able to use energy. Okay, so there are such molecules present that have motor activity, and those molecules are associated with microtubules. So there are two types of molecular motors associated with microtubules. One is the kinesin and the other is dynein. And the kinesins are observed to move from the minus end of the microtubule to the plus end, while dyneins are found to be moving or walking on the microtubules from the plus end to the minus end. So the kinesins and the dyneins both have a complex structure, quaternary structures. So as you can see here, the kinesin basically has two heavy chains, and the stalk region is a coil structure. So there are two polypeptides alpha helical that are coiling around each other to form a coil structure. It has the head domain. This is the head, and the head domain is the motor domain. So it has the binding site for GTP. So you must understand kinesins have a nucleotide binding domain, but they can bind to, they bind to ATP. I'm sorry. And you can see that the ATP, the motor domains have, or the head has motor activity. So they have inherent ATPase activity, and they can convert or hydrolyze ATP into ADP. And when they do so, energy is being released. That energy released allows it to walk. If you can see, this is the power stroke, this is recovery. When it puts the foot over, when it puts the head over here, that will be a power stroke, and then this fellow will recover. So it's almost like us walking on the roads, one step after the other. When we put one foot, that is a power stroke, and the other foot, when it is released, is a recovery stroke. When that foot is now put on the road, that becomes the power stroke, and that is how the molecular motors walk on the microtubules, which act as molecular tracks. Now, in case of dyneins, you have these as the motor heads. So this is a ring structure that is formed by several molecules that have ATPase activity, and this is the microtubule binding stalk. So these are the ones that are going to bind to the microtubules, and this is going to expend energy to have these bind to the heterodimers that are present on the microtubules. So, although we understand that kinesins move from minus to plus, so minus to plus is always called as anterograde direction, while plus to minus is retrograde direction. So, dyneins move retrograde and kinesins move anterograde. There are exceptions to rules. There are certain kinesins that are able to move in both directions, that is anterograde as well as retrograde. So, you will find a lot of molecular motors with exceptions to the rules. So as you can see over here, you have the heavy chains forming the head, stalk, and the tail, and the light chains are associated with the tail region. This is the region to which a molecule can, or a cargo protein can bind, or an organelle can bind. So as you can see over here, the globular heads will gradually, put a foot. Okay, that will bind to ATP. So the ATP bound to the motor domain is able to bind to the microtubule at at one of the tubulins. So alpha beta, if this is alpha beta, it will bind. Then the one that has left released itself is bound to ADP. When it binds to the next heterodimer over here, it will release the ADP, and then to this the ATP will be bound. At the same time, here the ATP is going to be hydrolyzed to release the ADP. So this will, when it is bound to ADP, it will remove itself from the heterodimer. So please understand that when the motor domain is bound to ATP, it can bind to the microtubule. Okay, but when the ATP is hydrolyzed to ADP, then it will remove itself from the, that is how one motor domain when bound to ATP will bind to the microtubule, and the other one which is expended, that is going to leave the microtubule and come to bind over here. So that is one step after the other. You have the motion happening. So this is power stroke and this is recovery stroke, and that is how each step is 8 nanometer, and interestingly, alpha beta tubulin is 8 nanometer long. So with every step, 8 nanometer region is being covered, and so for every 8 nanometer step, you have one molecule of ATP being used. So it is expensive, but it is needed, and it is regulated thereby. So where is this? Again, let us go to the neuron as an example. You can see how you have dyneins carrying molecules from the plus end to the minus end, and how you have kinesins taking vesicles, etc., from the minus end to the plus end. So you have transport across the axon happening. In cell division, you can see how you have the plus ends present towards the membrane and the minus end towards the centrosome. When it has to divide, you have two pairs of centrosomes being formed, and then you can see how you have the spindle fibers being arranged to cover the entire cell. Okay, so this is how microtubules are functioning in various specific cells to give it different functions. So let us make the conclusions. Microtubules are hollow cylinders made out of 13 protofilaments, with each protofilament made up of dimers made of alpha and beta tubulins. The microtubules have a polarized structure, with more dimers being added at the plus end of the microtubule and the minus end points mainly towards the MTOCs. The molecular motors, kinesin and dyneins, are able to move on the microtubules, carrying cargo using energy per step that they take. Okay. The microtubules are able to polymerize and depolymerize, leading to the phenomenon of dynamic instability, which in turn enables specialized functions such as in cell division. So microtubules as hollow cylinders are able to provide strong support as skeleton to the cell. However, that they can also lay down as railway tracks to allow cargo to be delivered from one station to another station inside the cell is intriguing in itself. Thank you.